Okay, so update. I just posted a YouTube video. It was like a help session for that quiz. It was atrocious. Not up to par with my standards of what I was trying to create. It was not fluid. So I found an easier way to do this using my laptop screen as a video input. Anyways, I'm not going to go into those details for you. Just know that this video is the same purpose as helping you with that quiz. There's some common mistakes I'm seeing, and actually I made a mistake too in that YouTube video I just sent out. It wasn't necessarily a mistake, I just approached it the wrong way. And so I want to make it real clear exactly what I'm expecting on that quiz for everybody. So hopefully you watch this before you try it and don't have to redo this. And if you do, no big deal. Hopefully I've got the settings set up so that you can unsubmit and then resubmit a document. I don't think when you turn something in, it's just a once and for all. If it is, I need to fix the settings so that you can take stuff out and resubmit. Or you can always add a new document and say, here's the new document, because sometimes you can just add more than one document. So I don't know, we'll figure that out. But point is, you got plenty of time to do this quiz and get it right. And if you do submit it, I'll let you know it's wrong and you should resubmit it to get it 100%. Because it's worth 20 points and it's going in the quiz category where there's not much there already. And so I'm trying to help out with that category. This is an open book quiz. In fact, you're going to need the book. You're going to need the tool that you got to use that's in the book. And so, anyways, I put that on Google Classroom. I also put it, I put it on Google Classroom, and I put it in this document here. I think I can do it to the quiz too if it's not too late for those of you who are already probably in the middle of it right now. So I need to move on then. Let's move quick. So let me go to my screen. Hopefully you can see this. Probably see a weird tunneling thing happen there, but now here is my laptop screen. Hopefully this is clear and everything. Hopefully you can see my mouse and everything, because I want to use it as a pointer. So hopefully, I think this is going to go way faster than my last video. This codon wheel, I might put this image in the Google Doc so that you guys that go open that doc after I do that, then you can actually have this in front of you. You should have it anyways in your textbook or in the keynote slide that I posted. It has all the pages of the textbook. That's another place you can find it. I even put a picture, straight up just picture that you can print, so you can just have a one-page deal. But I added it to the front of this quiz here, so I'm taking kind of the same quiz, but yet these are just different examples. And I'm using this picture just right above it so I can quickly scroll up. It's cool because I can point, I can use my pointer to show you how to use this wheel. So let's, without further ado, move down into this problem. So first thing to point out is that this is DNA. This is a DNA molecule. I know it's just letters to you probably, but hopefully you have watched the other two videos or have a strong understanding of DNA structure and nucleotide structure, so that these are not just letters to you and you kind of understand what's going on. Because remember, DNA structure, there's a lot going on there, but these letters just simply represent the nitrogenous bases in the nucleotides. And you remember the base pairing rules that thymine always pairs with adenine and cytosine always pairs with guanine. And so what you're seeing here is representative of a DNA molecule. A DNA molecule is two-stranded, right? Here's the top strand, here's the bottom strand. There's not really a top and bottom to DNA, but there is there are two strands, and if we arrange them like this, we can talk about it being top and bottom, so I'll use those words. But most important is these words: template strand, template strand. That's the most important strand, is the template strand, because that's what the RNA polymerase, the ribosome, is going to read. The ribosome is going to, it's RNA polymerase. Yeah, we're doing it all here. We're doing transcription and translation. The first thing we need to do is make a transcript, an mRNA transcript, which is done using an enzyme called RNA polymerase. He comes in here and he unzips the DNA and he reads the nucleotides one at a time and makes his own strand of RNA by grabbing nucleotides and putting them together. So when RNA polymerase sees a T, let's go ahead and get started. When RNA polymerase sees a T, he will put an A. When RNA polymerase sees an A, he will put not a T, a U, because RNA doesn't have thymine. It only has uracil instead. or it just has uracil instead. Okay, so the next one, we see C, we put G. We see G, we put C. So I'm just, oops, let me space. So I'm using base pairing rules here, right? And so let me finish this so that I can go quick. GG. Hey, again, when I see A, I'm not going to put T, I'm going to put U. Okay, you see what I did there? It's so amazing how your body does this. Your cells do this like at lightning speeds, and it just took me forever to do that. So it's coding, right? We've basically made a copy of the template strand, but it's not an identical copy, right? This, oh, got a little crazy there. Whoa, this strand is not an exact. The template strand was used as a template, and our mRNA strand is not an exact copy of it. Our mRNA strand is essentially a copy. It's kind of a copy because we used base pairing rules to read the template strand. So it's essentially the same information. This information was used to code for this information, right? And so we have to use the template strand always. In the DNA, you have this complementary strand, right? Just coincidentally, and it looks a lot like the RNA strand, and it should, because we use base pairing rules the same way, right? But the problem is we don't have thymine, we have uracil, and so it's not a good habit to just copy the complementary strand. A lot of students do that. They'll say, well, it's the same thing, so can I just copy that down? But then you forget to put uracil instead of thymine, or you get in the habit of thinking RNA polymerase somehow reads that, but it doesn't. RNA polymerase reads the template strand, and it reads each nucleotide one at a time, and as it does that, it adds the corresponding complementary base down here, and then makes a single strand of RNA, which will leave the nucleus, go into the ribosome, and be translated. And when it's translated, it's important to see that, let me make sure. Yes, okay. It's important that the that the ribosome knows to start reading this mRNA at the right spot. And where that is wherever you see AUG. So out of all the different amino acids, the 20 amino acids that are surrounding here, the wheel, my perimeter, and all the different codons that code for the amino acids, there's only one codon, and it's AUG, that codes for methionine, that is called the start codon. It's called the start codon. And it codes for a very special amino acid, methionine. Nowhere else on here do you see methionine. And methionine is always the first amino acid in a polypeptide. So we'll get to that in a minute. But for now, we just need to know that we can't start putting down codons until we finally see AUG. There's where we can start. Okay, so then you can do a space, or you can do a dash. Let's do a dash. We have AUG, CC. I'm just basically copying it down. I'm just copying it down, but I'm putting them like one at a time. I wonder if it would be better if we use a different character instead of a dash. Might be helpful if we put like a slash instead. Slash GGA. We do caps lock here. GGA. And then UAU. And then GCG. And then AUG again. So just because you have methionine as a start doesn't mean that's the only place you can have methionine. You can have it elsewhere. But once you have methionine, the ribosome is going to start assembling polypeptides. Then we have AUG again. And then we have AGU. Okay, so the anticodons. The anticodons are what are found on the tRNA. So let me just, I know I don't want this to be a major lecture video, but transcription, translation, I don't want that. I'll look at my images. I just want to show you something real quick. Here's a good one. You see, this here is the tRNA. And the tRNA is what's bringing the amino acid. You see it up there at the top? That's methionine. You see how it sees AUG as a start codon? Well, the UAC that's on the tRNA is the anticodon. So the anticodon is found on the tRNA, and it base pairs with the codons on the mRNA. Does that make sense? And so, but when you're translating, you don't look at the anticodon to see to use that wheel. You don't use that wheel. That's a codon wheel, not an anticodon wheel. But a lot of students think that, well, I'm looking for the right amino acid, so I'm just going to look here to see what the anticodon is and look at that and use that on the wheel. But that's wrong. It's the codons on the mRNA that you use in that wheel to figure out what amino acid will be brought. Now the amino acid will be brought only if this anticodon base pairs with the regular codon. Okay, anticodon base pairs with the start codon. Anticodon base pairs with the codon. And in this case, the start codon is always AUG. It's always AUG. And so, oops. In this case, that's what we have right away. Now, in the other examples, I think on the quiz, that's not the first codon right away. So you need to wait, you need to keep reading it until you see AUG, and that's when you're going to start putting the sequence down. Hopefully the next example lets us do that. So what are the anticodons here? It's going to be U, oh wait, I'm in caps already. U, A, C, GGG, CCU, AUA, and then CGC, and then UAC, and then UAC again, and then UCA. Okay, those are the anticodons found on the tRNA. Now, what amino acids do we have? What amino acids are we going to have? Well, don't look at the anticodon and come up here and try to use this codon wheel. It's a codon wheel, so we have to look at the codons. And AUG, again, August. Here's a way to remember this, guys. School starts in August, right? Well, the ribosome doesn't start reading this mRNA until it sees AUG, August, right? That's when protein synthesis starts, in August, AUG. And it always codes for AUG, always codes for methionine. So we write methionine. Methionine. And we'll just put a dash. What's the next one? The next codon is CCC. Come up here, I go. And remember, here's how you use this wheel. You start on the inside. It's one of these four. Whichever one you pick, then you got to go to the next four. It's one of these four. And then whichever one you pick, it's one of these four, right? Well, right now we have CCC. And so it's C, C, C. Proline. Okay, what's the next one? GGA. So we got G, G, A. That's glycine up there. Glycine. Okay, what's the next one? We got what was that, GGA? Yeah, GGA. Okay, that was right here. Now we got UAU. So what is UAU? UAU, tyrosine. Okay. What's the next one? GCG. So GCG, that's alanine. Then we got AUG again, that's methionine. And then AUG again, that's methionine. And then we got AGU. AGU, that's serine. Serine. And that's it. Now we should have run into a stop codon, I feel like. So I don't know if I made a mistake. I can double check if I made a mistake. I could have done this again. You guys, it's real prone, I am at least, to making mistakes here. I don't know what it is, but you know, if you're not a good speller or you make little typing errors, then it's real easy to miss one of these nucleotides and put the wrong letter. And I very well could have done that. That's called a mutation. That's actually one of our next lessons. When the body does that, it has a way of auto-correcting. Just like I just auto-corrected the anticodon, I spelled it anitcodon, and I just hit the autocorrect. Your body has a way of doing that too. So I'll use that as an analogy in that lesson. But in the meantime, right now, let's just focus on this problem, and we'll just say we don't have a stop codon. Maybe I didn't make a mistake. And so I actually don't have an answer key for this, but I'll double check it. Let's do this one now instead, just to see if we can run into a stop codon. The stop codons, by the way, are here. It's UGA. So anytime you see UGA, it means stop. Don't make any more. Even though there's plenty of more nucleotides left, you're not allowed to make any more amino acids. Everything falls apart. The ribosome falls apart, and then protein synthesis stops. But there's two other codons, UAA and UAG. I guess we didn't have any of those, did we? UAA or UAG? Yeah, we didn't have any of those. Alright, anyways, so I don't know actually if I do need to do this problem. It's kind of the same thing as the other one, but just so you know, we have the template strand here on this one on the bottom. So if the template strand's on the bottom, that's the one we want to read. We don't want to try to get in the habit of always reading the bottom one or the top one, because it depends on where the template strand is. I think up here it was on the top. That's the one we read. That's the one we read. We pretended we were RNA polymerase and we read that strand. And then if we read it, I'm just double checking myself here that I read this correctly. And it looks like I did. Yep, okay. So down here, we want to read the bottom strand that I just had highlighted. So to do that, we're going to have C, maybe right underneath it here, A, U, G. Ooh, this is not going to be right underneath. Maybe it's because I didn't space or something. We got C. It's okay, it doesn't have to be right underneath. It's easier for my eyes though. We have C. Now we're looking at a T, so we need an A. Now we're looking at an A, so we need a U. Now we're looking at a C, so we need a G. Now we're looking at a G, so we need a C. Now I'm just going to continue the process without saying it. Okay, so there's my mRNA strand. Now you might be tempted to think, and this is what I did in the last video, my first codon would just be CAU, but it's actually not, because the RNA polymerase is just going to go do and travel along the mRNA until it finally reaches AUG, which is real quick. It's right here. And so let's start there. Let's say that that's our first. Let's say that that's our first codon. So our first codon will be AUG. Put a dash, or we can just do a space. AUG, and then we have CGC. I'm just trying to make it a little easier to read on the eye, so we can actually put a couple spaces. AUG, CGC, AUA, UGG, CUG, UAA, and then G. So that doesn't even make up a codon, right? So that's, hopefully that means stop, and I think it does. The anticodons, we can go back and do that later. Right now we have the codons. We have the codons which are used in this wheel to figure out our amino acids. And we know AUG is methionine, because if you look up here, AUG is methionine. That's that start codon that codes for the first amino acid. So our first amino acid is methionine. And then the next one is CGC. So I do CGC, that's arginine. And then the next one is AUA. And so that is AUA, that's isoleucine. Isoleucine. And then the next one is UGG, and so that one's going to be UGG, tryptophan, and the next one is CUG, that's leucine, and then the next one is UAA right here, and UAA, boom, UAA stands for stop. And so I know we're kind of at the end of the thing anyway, but that means stop. And I'm okay if you put stop, and I won't count you off. But technically, everything, the tRNA that comes here and attaches, it'll attach, but it doesn't bring an amino acid. It doesn't drop off an amino acid. It just comes and then brings something else. It brings some other molecule that allows the whole process to stop. It allows the ribosome to disassemble into its subunits, and the polypeptide chain will be released into the cytoplasm to go through the Golgi apparatus and be packaged and shipped. Remember, the Golgi apparatus is the post office for proteins, and so the Golgi apparatus will do more sorting and folding and twisting of the protein to finally shape it. Proteins are all about their shape, and right now we're just making the amino acid sequence, which is the primary structure of a protein. So it's pretty important, because this structure, this sequence, is going to dictate how it ends up being folded and twisted later on. And so you can put stop there, you could just leave it blank. You technically have, because it's not actually an amino acid. So there you go, there's your amino acids. You have a start, always methionine. So I noticed, I mean, if you are one letter off, if we started with our codons as CAU, and then GCG, and then CAU, we'd have a totally different sequence of amino acids. You have to be careful. And then for your anticodons, well, it's just base pairing with the codons. The anticodons on the tRNA are going to be just opposite. I don't know how you want to say it. Complementary is how you say it, actually. Complementary to the codons. GCG, UAU, ACC. You get the point. The last thing I wanted to help you with, I don't have an example here, but maybe I can just work through one real quick, is if you're dealing with a sequence of amino acids and you're working backwards to DNA, how do you do that? Let me show you how to do that. We'll just do a quick one. Let's say we're doing methionine, lysine, and glutamine, right, and so on. I'm just going to do 3 so we can make it quick. These are the amino acid sequence. So let's say we started here, and I wanted to know the DNA. So I'm going to scroll down. It's going to take a little bit of work to get here. So DNA strand. DNA is not just one strand. So at some point we're going to have eventually a DNA template strand, and then we're also going to have the DNA just the complementary strand. Alright, and so that's our ultimate goal, is to come up with some sequence, the 2 sequences of nucleotides that make up the 2 strands of DNA. So how do I get to that point from just starting with amino acids? Well, what I'm going to do is I'm first going to figure out what were the codons that coded for these. What were the codons that coded for these? Well, methionine we know is AUG, right? Lysine, check it out. Lysine, you have some options. It could be AAA, could be AAG. Let's just say AAA, AAA. Let's make this easy. So you see how you have options here? I'll have to be careful when I grade your stuff. Lysine, it could be, let's see, where's lysine? Here it is. It could be AAA. Wait, what? No, we just did lysine. Sorry. Glutamine, the third one. Glutamine, and don't mistake that one for glutamic acid. That's actually a different one. Glutamine is down here, and it's CAA or CAG. Let's say CAG. That's the one I want to do, just because. CAG. All right, that we should label this is the mRNA sequence. In fact, those are the, let's say this, the mRNA codons. Right, those are the mRNA codons. And so what does the mRNA strand look like? Well, it's just essentially all of that. Right, so do this. Here, copy, paste, and maybe clean it up a little so it's evenly spaced. Okay, so if that's our mRNA strand, this would be our mRNA mRNA strand. Then what would the template strand of the DNA look like? Well, and let me just bring this down so it's a little easier to see. What would the DNA template strand look like? Because remember, the template strand is what's used to code for that, and it's following base pairing rules. So if adenine is on the mRNA strand, then on the template strand we had what? Thymine. If we have uracil, what base pairs with uracil? Adenine, right? What base pairs with guanine? Cytosine. What base pairs with adenine? Thymine. We got that 3 times. And then what base pairs with cytosine, guanine? You get the point. So I can continue on there. There's my template strand. Now what does my complementary strand look like? Well, you could actually do, some of you may have saw me do this. I'm going to get a little lazy too here. I'm going to copy the mRNA strand because it essentially is the same, but we do not have uracil in DNA. So take all your U's and replace them with T's, not Y's, sorry, T's. Replace, wait a minute. That was the only U, huh? There you go. There you have it. Here's your DNA molecule. Hopefully that helps you guys. I'm going to cut to a regular screen here. Oh, sorry. I think I did. Whoa. I'm getting this technology thing figured out, I think, hopefully. And so this stuff is tricky, I know, and it gives me a headache just doing it too, because it's a lot of like reading and like little characters, and it's just, if you're one letter off, it's all wrong. And don't get stressed out. If you do, just start over. I tried to make it simple. It's just two problems, but I know you have the RNA and you have the DNA and you have the amino acids. It's a lot of work. On the test, I'll have minimal as well, but enough that I can see you go from the DNA all the way to the amino acids, or from the amino acids back to the DNA. Either way, either direction, you should be able to do that. All right. So hopefully that video helps you. If you need to fix your quiz, do so. That'll hopefully boost everybody's quiz grade a little bit, because that's 15% of your overall grade. And you got plenty of time till May 1st, I think, to do that, up until the test day to take this quiz. All right. Adios.